Hello, everyone, and welcome to this 8th episode of my publication at a Glance. Today, we welcome Dr. Emanuela Berrino, who is a postdoc at the CEA in Saclay, Paris. Emanuela started her career completing her PhD at the University of Florence. She then pursued a postdoc at the University of Sapienza in Rome. And she then integrated the group of Professor Sébastien Thibaudeau at the University of Poitiers. Today, she will talk to us about her research at the University of Poitiers in collaboration with the University of Florence about the discovery and development of HCA4 inhibitors enabled by superacid chemistry. Emanuela, the floor is yours, and thank you for joining us.
Thank you very much for the introduction and for giving me the opportunity to present this project, that led to the identification of selective and potent carbonic anhydrase 4 inhibitors. Carbonic anhydrase is a metalloenzyme that catalyzes a simple but crucial reaction for cells. That is the reversible hydration of carbon dioxide to generate bicarbonate and protons. These species are involved in a plethora of biochemical reactions, and for this reason, abnormal activities or level of some carbonic anhydrases have been associated with diseases, leading to the development of inhibitors for their treatment. However, despite massive efforts in the development of these inhibitors, very few of them are endowed with a very strong selectivity for one isoform over the others. One isoform in particular attracts our interest, carbonic anhydrase 4, because for this isoform, selectivity still represents a complicated issue. CA4 is one of the 4 membrane-bound isoforms, and although being known for a long time, it has not been yet validated as a target for any disease. This was probably due for the lack of potent and selective inhibitors targeting these isoforms. Also because the few reported compounds targeting CA4 belong all to the primary sulfonamide class, which is the most employed, but also the least selective among the CA inhibitors. For this reason, in our project, we want to increase the inhibitor selectivity index, enlarging the chemical diversity among the new CA inhibitors to be developed. For this reason, we started from this compound previously reported in the Thibaudeau group that showed a very nice potency and selectivity against CA4. To the secondary benzene sulfonamide bearing a propyl amino chain with a fluorine atom. Starting from this building block, we designed this library of compounds, all containing non-primary sulfonamide moieties and this fluorinated propylamino core, also considering the importance of fluorine in medicinal chemistry. Moreover, polar chain were inserted bearing a primary amine function, considering that the entrance of the active site of CA4 is rich in polar and charged amino acidic residues, so that this could drive the preferential entrance of the compounds in the active site. For the synthesis, we started with the preparation of these fluorinated building blocks generated in superacid. Superacid is a specialty at the IT2MP of Poitiers, and through the presence of HF and SPF5 system, it allowed the generation of these super electrophilic species that are able to react with very poor nucleophiles, allowing to generate compounds that cannot easily be obtained using classical synthetic procedures. With these compounds in our hand, we tested them against 4 carbonic anhydrase isoforms, using the stopped flow CO2 hydrase assay. And as you can see, the most polar compounds bearing this primary amino tail and the propylamino core show it to be the most potent and selective among the series. Moreover, the presence of fluorine revealed to be very important to increase selectivity over CA12, which is another membrane-bound isoform often co-expressed with CA4. To understand the binding mode of this compound, we performed molecular modeling studies, and the results suggested that this compound inhibits CA anchoring to the zinc-bound water molecules, blocking the catalytic cycle. The propylamino tail is oriented in the hydrophobic pocket of the active site, whereas the primary amino group in the polar chain is oriented through the hydrophilic pocket, being stabilized by an interaction with the proline 201. Conformational analysis were also conducted within the active site, showing that the fluorine insertion is very important to stabilize the orientation of the polar chain towards the hydrophilic pocket. And this stabilization, in fact, is missing in the non-fluorinated analogs. In conclusion, we believe that these compounds can represent a very important tool to further understand the physiopathological roles of carbonic anhydrase 4 with the aim to possibly validate these isoforms as a therapeutic target. If you want more details about this work, please check the publications, and of course, don't hesitate to contact us if you have any more questions. Thank you.
Thank you very much, Emanuela, for this very interesting presentation and for sharing your results. To anyone watching this video, if you're interested in showcasing your results in the same way, do not hesitate to contact us. You have all the contacts on that slide. And thank you. See you next time. Goodbye.
Bye.